Hello and welcome to another lecture from Mr. Watkins. This particular lecture is on action potentials, and specifically we're going to look at the action potential down the axon of a neuron. Before we get going on that particular piece, we just need to make sure we review some basic biology, and we're going to do that through how this neuron and how this membrane is going to act during an action potential. So I'm going to go ahead and draw a cell here. The black line here represents the cell membrane. Now remember, the cell membrane is a structure that basically walls off, if you will, the inside from the outside of the cell. It controls the materials that go through the membrane so that way the cell on the inside can have a different electric potential, have different ions, have different molecules on the inside than compared to outside. We also need to discuss this thing called diffusion, something that you should have had back in biology. Basically, it's a material or particles that are in a high concentration will tend to move to a lower concentration. So for a neuron, for an axon, or most cells, typically they are in a solution. The outside of the cell has a lot of sodium ions in it, usually because of sodium chloride, and we have the salt that dissociates in water. The concentration of the sodium outside is higher than the inside, and so therefore the sodium wants to move inside the cell. Also, inside the cell, there are potassium ions, and so we have a few of these positive charges, but we also have some proteins inside the cell that are negatively charged. And if you remember from your junior high science, positive charges are attracted to negative charges. And so there's also, the sodium is also attracted electrically to the negative proteins on the inside of the cell. Now, they cannot move because the cell membrane is selectively permeable, and it doesn't allow the sodium ions to diffuse through the cell. Now, there is some leakage, a very small amount of leakage, but it's very, very small. Now, the concentration of the potassium ions and the negative charged proteins inside the cell is high when you compare that to outside the cell. The potassium is also attracted to these negative proteins, but the proteins can't leave the cell because the cell membrane prevents them from leaving. The potassium does want to move outside the cell because, again, the amount of potassium ions inside the cell here is a lot higher than outside the cell. And so diffusion is basically saying that they want to go to the outside. Now, the cell, the membrane, does have a special channel protein called a potassium leak protein, or a potassium leak channel, and I'm just going to draw that with a little line here. And what that does is it allows some of these potassium to move outside the cell. It is always open during the action potential. So think of it like a boat with a small leak in the bottom of it. You're always going to have a little bit of water moving into the boat. In this case, the potassium ions are moving out, and the area outside the cell becomes more positive. Now, the separation of charges here, it creates a potential difference because we do still have a lot more positive charges than we do outside than we do on the inside. There is a difference, and we measure that difference in something called volts. And so you probably have heard of that. Now, for cells, because they're so small, we measure it in what we call millivolts. For most neurons at rest, the difference, and by at rest I mean the neuron is not doing any work, is not going under through a bunch of activities, is not, in this case, doing an action potential. So when the neuron's at rest, typically they have a potential difference. They have volts of negative 70 millivolts, and we use the negative to indicate that just inside the membrane it is negatively charged compared with outside the membrane. Now, there are some other special proteins that are found in the membrane, and these are called voltage-regulated channel proteins. And I'm going to draw one down here, and I'm just going to draw that with a little cap on the top of it. And these are voltage regulated channel proteins. And what these do is when a certain voltage, a certain difference is met at the membrane, they open. So there's a there's a voltage regulated sodium channel protein, and there is one that's for potassium. The sodium one actually will open and allow sodium in when the difference hits about -70 to -55 millivolts. Okay, so let's look at how all of this relates to an action potential. So I'm going to draw an action potential graph here. I'm just going to hand draw it. And we've got our axes here. So this is our y-axis, and this is going to be voltage, and it's in millivolts. And then about here, I'm going to draw a line across. This will represent 0 millivolts. And then this will be time, and I'm not going to write time right there in the middle, so because we're going to put the picture here as we go through. Now down here towards the bottom, I'm going to mark this at -70 millivolts. And if you remember, the neuron at rest has a resting potential of -70 millivolts. Now what's going on is, so as time goes by, the neuron is trying to maintain this resting potential, and I'll do this by drawing this line here, this resting potential of -70 millivolts, meaning the difference between the positive charges outside and the positive charges compared to the positive charge inside is -70. Now what's going on at this time in the cell is that we have our potassium leak channels are open. They are always open through this entire thing, and so the potassium's leaking out. And then to help kind of maintain that, we have this special sodium potassium pump that is working, and we'll talk more about that as we get further into this. So here is the cell. It's at rest. It's not doing a lot of activities, but let's say that some stimulus occurs, maybe it occurs and the dendrites pick that up, and we get down into a summation where we're getting ready to have an action potential happen. Some stimulus occurs, and that stimulus occurs causing some sodium to leak in, and it has to leak in above what we call the threshold value. And the threshold value typically for a neuron is -60 millivolts to -55 millivolts. Once that occurs, once we hit that threshold, the stimulus causes the threshold level to be reached, we have a dramatic, huge increase in the amount of sodium ions that rush in, all the way up to a +30 millivolts. It causes a +30 millivolts difference. We have so many ions that rush in. Well, what happened here? At -70 millivolts, the voltage regulated sodium channel opens. So the voltage regulated sodium channel opens. Now remember that the concentration of sodium outside the cell is higher than inside the cell, and so diffusion says it wants to move in. It's a lot higher. It really wants to get in. Also, the amount of positive charges is greater outside the cell than inside the cell. And this electrochemical difference really causes the sodium to want to move in quickly, and it changes that potential difference, changes it very quickly in a millisecond or mere milliseconds. And we call this depolarization, this process by which the membrane potential changes. When it moves from this -70 towards 0, that is depolarization. Now, again, think about it this way. It was polar because we had a higher amount of positive charges outside than compared to inside, so there was a difference. It was polar. Once this voltage-regulated sodium channel, once the difference hits close to a +30 millivolts, that causes this voltage-regulated channel to close. And what happens is that happens to be about where the voltage regulated, my pen just broke, the voltage regulated potassium channel opens. So again, it's voltage regulated. So the idea here is that when the electrochemical gradient, when the potential changes, it's going to cause this channel protein to open, and when it does, the potassium then rushes out. And if you'll remember, there was more potassium on the inside than there was on the outside, so diffusion is going to have it move out. Also, because all of these sodium ions moved inside the cell, it's more positive, and so the inside of the cell is more positive. Remember, it's plus 30 millivolts. It's going to want to go to the more negative outside, or the less positive outside, which seems to be more negative, outside the cell. So we have, again, that electrochemical difference causing it to move. And it will move quickly as well. And so the potassium ions move in. And we get down to about, well, as it moves in, we call this repolarization. We are going from having it not have as big a difference back to having a more polar or a bigger difference between what's inside and outside. Now, excuse me, as this occurs, as the amount of potassium comes in, and we get close to this negative 70, that causes this voltage-regulated potassium channel to close. And as it gets close, it starts to close. But it takes about a millisecond for it to close. And in that time, when it's down here about at resting potential, the time it takes for it to close, more potassium leak out. And we actually even go even lower. And we can go down to as far as negative 90 millivolts. About 90 millivolts is where we bottom out on this. And so we go down so far, by the time the closing happens at negative 70 millivolts and the amount of potassium that leaks out as the thing is closing, we get what's called hyperpolarization. So basically, we've now overshot, if you will, what our resting potential needs to be. And so what brings it back to this resting potential, back to being like it was before, is because both the sodium channel and the potassium channel are now closed. But remember, these potassium leak channels are open, and so we get some potassium to leak out. And then the sodium and the potassium pump also begin to work, and it gets regulated until it gets back up to its resting potential at negative 70 millivolts. So let's talk about that sodium-potassium pump. That sodium-potassium pump requires ATP. And so at this point, the sodium-potassium pump is working hard along with the potassium to help reinstate that resting potential. The sodium-potassium pump, what it does is it pumps inside 2 potassium ions, and it pumps out 3 sodium ions. So you can see that we're pumping out more positive charges than we're pumping in. And between the leak from the potassium leak channel and the pump working to bring in or get rid of 3 charges and only bringing in 2, we get this hyperpolarization back. Notice that I said that this was a pump. Excuse me. And so this pump requires ATP. It requires ATP to work. So this leak channel here, this is all basically facilitated diffusion. There's no energy being used. This voltage-regulated sodium channel, again, is facilitated diffusion. There's no ATP being used to cause this to open. It's all based on voltage, on the difference in potential. The same with this potassium voltage-regulated channel. And so we have to, the only thing that's active transport, and it's what's used to help maintain the cell, is this sodium-potassium pump, and it uses ATP. This is basically how it works. We end up having the sodium channels open, sodium rushes in, changes the potential on the inside of the cell, right on the inside where that membrane is. We get to a certain voltage, usually about 30 millivolts. That causes those channels to close, the potassium channels to open, and we repolarize that membrane. And that repolarization occurs until we get down to about negative 70 millivolts. When this one closes, it just overshoots a little and we get some hyperpolarization. And to restore that back to the resting potential, the cell then uses those leak channels and the sodium and the potassium pump to help bring it back and get ready for the next stimulus that comes through. It also should be noted that if another stimulus tries to come in during any of this time, it's not going to have any effect, or not going to have an effect, because we've already started this process. This process takes milliseconds to occur, and it goes right down the membrane, and that's how that signal travels. If you have any questions, just see me in class.